Every living eukaryotic cell contains an extraordinary organelle called the Golgi apparatus. It acts as the cell's processing, ensuring proteins and lipids reach their correct destinations. The Golgi complex was discovered in 1898 by the Italian scientist Camillo Golgi. Silver staining technique, complex membranous structure that later became one of the most important discoveries in cell biology. Golgi apparatus originates from transportals that continuously bud from the rough endoplasmic reticulum. These vesicles fuse together and maintaining the dynamic structure of the Golgi complex. The Golgi apparatus is found only in eukaryotic cells. It is usually located close to the nucleus, contain fewer but larger Golgi complexes. The Golgi apparatus consists of flattened membrane sacs called cisternae, interconnected tubules, vesicles, and vacuoles cellular products. Cisternaes are flattened membrane-bound sacs arranged in parallel stacks. Each cisterna contains specific enzymes and lipids as they travel from the cis phase to the trans phase. Tubules are thin membrane channels connecting different Golgi compartments. They facilitate rapid transport of membrane materials and selected proteins between adjacent cisternae. Golgi vacuoles temporarily store processed proteins, lipids, and other cellular products before packaging them into transport vesicles for delivery throughout the cell. Golgi vesicles are small, spherical, single membrane-bound sacs that transport proteins and lipids within the cell. They occur as transitional, secretory, or clathrin-coated vesicles. Transmissional vesicles transport newly synthesized proteins and lipids from the endoplasmic reticulum to the cis face of the Golgi apparatus, initiating their processing journey. Secretory vesicles bud from the trans face of the Golgi and transport finished products to the cell membrane, where they are released outside the cell through exocytosis. Clathrin-coated vesicles possess a specialized protein coat that helps package selected molecules and delivers digestive enzymes to lysosomes with remarkable precision. The cis face is the receiving side of the Golgi apparatus. Newly synthesized proteins and lipids produced in the endoplasmic retic arrive here inside transport vesicles for further processing. As proteins move through the medial cisternae, specialized enzymes modify them by adding or removing carbohydrate chains. Phosphate groups are essential for proper protein function. The transphase acts as the shipping department. Here, proteins are sorted, packaged into vesicles, and directed toward lysosomes, the plasma membrane, or outside the cell through secretion. The Golgi apparatus is the cell's central processing and distribution center. It modifies, sorts, packages, and directs proteins to their correct destinations. The Golgi apparatus absorbs proteins, lipids, carbohydrates, and other compounds from transport vesicles, preparing them for chemical modification and intracellular distribution. One of the Golgi apparatus's most important functions is the formation of secretory vesicles that safely transport hormones, enzymes, antibodies, and neurotransmitters. The Golgi modifies newly synthesized enzymes through glycosylation and other chemical reactions, producing fully functional enzymes capable of performing specialized cellular activities. The Golgi temporarily stores processed proteins, allowing the cell to regulate when and where these proteins are transported or secreted. During sperm maturation, the Golgi apparatus forms the acrosome, an enzyme-filled cap that enables sperm to penetrate the egg during fertilization. In certain specialized cells, the Golgi apparatus participates in forming intracellular crystalline structures by concentrating and organizing specific proteins. In hormone-producing cells, the Golgi apparatus packages peptide hormones into secretory granules, ensuring their safe storage and regulated release into the bloodstream. In plant cells, the Golgi apparatus synthesizes pectin and hemicellulose, essential components of the primary cell wall that provide strength, flexibility, and structural support. During plant cell division, the Golgi apparatus produces secretory vesicles filled with cell wall materials. These vesicles fuse at the cell's center to form the cell plate. The Golgi apparatus is the cell's chemical factory and global distribution center. By chemically modifying, quality checking, packaging, and directing proteins and lipids with remarkable precision, it coordinates secretion, growth, communication, defense, and cell wall formation, making it indispensable for the survival and organization of every eukaryotic cell.